Thank you very much for the introduction. Thanks to the organizers for the opportunity to present some of my research about which is a bit different for some of you who know other parts of my work of the core. I'm not going to talk about metastasis today, but another part which is of using mathematical modeling for helping to inform on the scheduling of combination treatments, and in this case of the scheduling between bevacizumab, which is an anti-angiogenic drug, with cytotoxic therapies. With a specific, I wanted to present to you a specific study on how to combine experimental data with mathematical modeling. So as we probably all know, angiogenesis tumor angiogenesis is a process by which primary tumor, or a tumor, not only a primary tumor, but a tumor grows by attracting nearby blood vessels and developing its own blood tumor vasculature, and it does so by releasing some molecules that were called tumor angiogenic factors by Judah Folkman when he proposed this concept for cancer therapy, and which were then discovered in the late 80s by Napoleone Ferrara as being a vascular endothelial growth factor. And it releases these molecules which then stimulate the proliferation and movement of endothelial cells that move towards the tumor and then develop a particular a blood vessel network. And so the strategy that was proposed by Judah Folkman was to target these factors in order to cut the support from blood vessels and then from oxygen and nutrients to starve the tumor. And we had to wait until 2004 to see the first drug that was approved the first anti-angiogenic drug that was approved, which is called bevacizumab. It's a monoclonal antibody that targets this molecule, VEGF. And the thing with, so it was approved, as I was telling you, in 2004, I think first in colorectal cancer. And the thing that is a bit noticeable, it's that it has only proved clinical efficacy when it's combined with cytotoxic, with classical chemotherapy. And yeah, so just as a quick advertisement, if you want to really know much more about the biological processes behind resistance to anti-angiogenic therapy, I strongly invite you to go to John Ebo's talk on Friday. But that's not the process I'm going to talk to you about today. It's the so-called normalization process, which was put forth by Rakesh Jain in the early 2000s, and which is a possible explanation for this success of anti-angiogenic therapy, but only when combined with cytotoxic drugs. And what's the idea behind this? The idea is that when you look at tumor vasculature, as here, where there is the tumor is here in the brain of mice, and when you look at the blood vessels, you can see that they are very poorly functional. They are leaky, they're tortuous, they're not functioning very well. So that's why tumors are highly hypoxic, because the nutrients, oxygen, are not reaching the tumor, but that's also why the drugs, the cytotoxics, chemotherapy cannot reach the inside of the tumor and is not as efficient as it could be. And somewhat paradoxically, what was first proposed as a hypothesis, but then mechanistically proven by the team of Rakesh Jain in the 2000s, is this normalization process where from an abnormal vasculature that generated by the tumor, when giving bevacizumab, then the vasculature gets normalized, which paradoxically for an anti-angiogenic drug improves the quality of the vasculature and improves possibly then the delivery of the cytotoxic drugs.
Is this what Jane called pruning?
Yes, that's pruning. Yeah, that's what he calls pruning. So realizing this suggests that there is an optimal time window for the improvement of the delivery of the cytotoxic drugs, which would be after this normalization phase or during this normalization window. However, as of today, bevacizumab is clinically administered concomitantly with the chemotherapy. So because it's more convenient in the clinic, the patients received the two drugs at the same time. So this leads the question that is there a possible better administration if we give the two drugs sequentially with one drug, and in this case the bevacizumab first, and then the cytotoxics? And then how modeling could help in this case is that, well, there are an infinite number of possibilities of gaps that can be tried experimentally. Not all of them can be tested because that's too many possibilities. So then we were wondering if we could build a mathematical model that could try all these possibilities in silico and test and suggest optimal time gap between the administration of the two drugs, and maybe also capture the inter-individual variability for designing personalized therapies. So that's the scheme of the study that we conducted with in collaboration, strong collaboration with a experimental therapeutics group in Marseille called the Smart C team, where with a constant back and forth between the mathematical modeling and experimental models, we used experimental data to calibrate models, then generate predictions and go back to the experiment. But first, actually, we first started this study by a theoretical work where we, back in 2012-13, we studied this process with a very large model with tons of equations, even more parameters, which could simulate the spatial structure of the tumor, spatial structure of the blood vessels. But the major drawback of this type of models is that it was completely impossible to confront to the actual experimental data. There is way too less measurements that allow to calibrate the parameters of these large-scale models. And so most of the parameters in this model are impossible to estimate, and this model can hardly be used for predictions of an optimal delay. So we went simplifying the model, and there is actually a very nice, much simpler model for anti-angiogenic therapy, which was proposed by Philip Hennenfent and the team of Judah Folkman back in 1999. Which consists in departing from the classical Gompertz model, where you have this K, which is the carrying capacity, the maximal size that the tumor can reach. And the great idea of this model is to consider that this K represents the vasculature and to make this vasculature being time dependent, having a dynamics, and these dynamics can be controlled by the balance between stimulation, endogenous stimulation by VEGF, and endogenous inhibition by some endogenous inhibitors. So in order to adapt this for the case of normalization, what we proposed was to adapt this model and to divide the carrying capacity, representing again supposedly the vasculature, into two compartments of an unstable compartment and a stable compartment with stable blood vessels and unstable blood vessels, and to define somewhat more heuristically some general quality of the vasculature as the ratio of the stable over the sum of stable plus unstable. And then, so when we simulate this, but with only arbitrary parameters of the model that were just fixed for the purpose of illustration, we can illustrate what would happen for tumor growth if we give first the anti-angiogenics and then the cytotoxic at a later time, after either 1 day, 2 days, 3 days. This is what I call the gap here in days. So after the end volume here in cells, but again, the parameters here were not calibrated from data. We can see the end the final volume and try to predict some optimal time gaps. But this again was not confronted to clinical data, to experimental data. An important component in this going towards the data is the pharmacokinetics of the agents. And as you will see in the next slide, the first data that we had was paclitaxel and bevacizumab in a breast cancer model. And so these are the pharmacokinetics of these two agents, and in the concomitant scheduling and the sequential scheduling retrieved, so with parameters retrieved from the literature. But what is important to notice here is that bevacizumab, as all antibodies, it has a very long half-life, whereas the cytotoxic, the Taxol here, has a very, very short half-life. So then we had our first experimental study with here in black the control tumors, in blue the concomitant administration of the two drugs in the cytotoxic, and in red the sequential administration of the two drugs with first the bevacizumab and then the Taxol. So we did observe a better result with the combination. And we could fit, so these are the plain lines are fits of the model to this data. So we could reproduce the data, we could fit the data with the model I was showing you before. However, when you look at the parameters estimates, you see that we have very large standard errors in the parameter estimates. So we had identifiability issues. The model was still too complex to be confronted to be reliably confronted to the data. So we even simplified, and that's actually the last round of simplification, but there was one round, then another round, then another round of going back and forth simplifications of the model to have the least possible number of parameters in order to be both to both fit the data and be identifiable. This is the last model where there is still the Gompers. We forgot about the dynamics on the K, and then just model this increase of the quality of the vasculature this way, with just a delay from the constant A. Here is the concentration of the anti-angiogenic agent. Then there is the cytotoxic, like the cells go from the cancer cells are affected by the cytotoxic, and then they go through these three compartments, and this is like some model that was validated by Simeoni and colleagues in 2004 for the effect of cytotoxic drugs.
So, wait, Sébastien, so you don't have dynamics for Q, yeah? You have...
Q, it depends on T. It still depends on T, but there's no equation on it.
Yeah, that's what
I mean, there's the equation. It depends on the pK of A, and the pK of A is a linear one compartment pK. So then we also wanted to move to a more clinically relevant experimental model where, because bevacizumab is actually not used in the clinic for treating breast cancer patients, it's not used anymore, but it is used to treat lung cancer patients. And in this experiment, then it was more or less the same. We had this human cancer cell line, and we were measuring the tumors by luminescence. These are the experimental results that we obtained. These are the tumor growth curves and the survival. And as you can see, this is with the combined, so this is with the concomitant scheduling, sequential scheduling, and the reverse sequential scheduling of the two drugs. We had an improvement both in terms of tumor size and in terms of survival when using the sequential scheduling of the two drugs. And to fit this data, but to improve again the robustness of the estimates and to potentiate all these population data in these groups, we used what is called the nonlinear mixed effects modeling framework, which is a population approach, which I will shortly describe now. And it consists in going away a bit from the classical framework for nonlinear regression. This classical framework usually considers each animal independently, and it fits each time series, each growth curve independently from each other. And so you have beta j, which would be the parameter you want to estimate, and when you apply maximum likelihood principle to this statistical model, then basically you end up with a least squares minimization problem. When you are going to wanting to pool all the data from a population together, then there is a very nice framework which is called again this nonlinear mixed effects, where it's more or less the same, but all these beta j's from all the animals, then you say all the animals belong to the same population, and you treat this population as having a distribution of the parameters in the population, and this distribution of the parameters then has only a limited number of parameters, for instance, a mean value and a variance. And so you only have to fit the mean value, you only have to identify the mean value and the variance across the population to describe your entire data set. So so instead of having p times n, where n is the number of animals and p is the number of parameters, to p times n parameters to estimate, you have only p plus p squared. And this is very useful, especially when you have sparse data. So these are the fits that we obtain in this framework. So these are all the dots are all the data pulled together for the control, simultaneous, sequential, reverse sequential and sequential. And as you can see, the model was able. So this is the median simulation of the model. This is the 10th and 90th percent confidence. Not confidence interval, but the intervals for the inter-animal variability that is estimated because you estimate the standard deviation of in your population. And as you can see, it captures both the general dynamics, and the broken line is the median of the data. So it captured both the general dynamics of the median and the variability. And this is a very important part of capturing the variability of the dynamics. These are individual fits that then can be performed. And as you can see, when we look at the model parameters, then the identifiability of the parameters quantified by the standard errors is much more improved, which means that we have a much better, robust identification of the parameters in this case. Then we can plot all the median curves together to just have a graph, because this essentially summarizes all this data into one plot, because these simulations are data informed, and you can see that as suggested by the data and as seen, the sequential scheduling again performs better. So once we have this model that was the model that was calibrated on the data, then we could run simulations again as a function of the gap, what would be the final or the area under the here is the area under the tumor growth curve. It's a measure of the efficacy for schedulings where we change the gap, the interval of administration between the two drugs. And here we identified that a 3 days interval would be the best, and an 8 days interval would not be as good. So then we went back to the bench and tested this experimentally. When we tested this experimentally, we did in a second round, so this is a second round of experiments that were not used to calibrate the model. And then in the second round of experiments, we indeed observed that a 3 days time interval was better than an 8 days sequence for administration of the two drugs. And last but not least, and I think this is a key point of the usefulness of the model in this case, is that since we have a quantification of the inter-animal or inter-subject variability, we have standard deviations on our parameters, so we can simulate the population. And among this population, it's not only the median that is important, it's the variability. And we can, since there is variability in these parameters, there is variability in the simulation, so there is variability in the optimal gap. And so this box plot here represents the variability of the optimal gap that we find when we simulate the distribution of the parameters. And you see that there are some outliers for which it would be interesting to identify in the clinic. So in conclusion, my main message, I think, here is that from personal experience in this study that actually was a bit long at the end of the day, but it's that in order to be confronted to empirical data and real robust predictions, mathematical models must remain simple and well adapted to the data that is available. Mathematical modeling can be used to identify optimized drug regimen for combination therapies among a large number of scenarios that cannot be all tested experimentally. This is of increasing relevance in modern oncology, where an always larger arsenal of anti-cancer agents becomes available to oncologists, and especially in the era of immune-oncology agents, which now will be used in combination with anti-angiogenic drugs, combination with cytotoxics, or in combination with radiotherapy. And I mentioned here a recent paper that we performed together with the SmartC team, and especially Raphaël Serre, who is the first author on this publication for modeling the combination of immune checkpoint inhibitors in synergy with radiotherapy. Nonlinear mixed effect model is a great tool, in my opinion, for addressing all these questions, and especially it can allow patient-specific Bayesian estimation of the parameters that can be used for personalized scheduling. With this, I would like to acknowledge all the people that were both on the clinical and experimental and mathematical side that contributed to the study, and thank you all for your attention.